Evo 2 unveils breakthrough, largest open-source AI model in biology predicts pathogenic gene mutations with 95% accuracy. Date, February 19, 2025. On February 19, 2025, a transformative advancement in the realm of biological sciences was announced as the ARC Institute, in partnership with NVIDIA and a consortium of researchers from Stanford University, UC Berkeley and UCSF, unveiled Evo 2, the largest open-source AI model in biology to date. This groundbreaking model, which has been trained on over 9.3 trillion nucleotides sourced from more than 128,000 genomes, encompasses a diverse array of lifeforms across all biological domains, including bacteria, archaea, and eukaryotes. Evo 2 represents a significant leap forward from its predecessor Evo 1, which was focused exclusively on single-cell genomes. Key features and capabilities of Evo 2. Evo 2 is designed to identify intricate patterns within gene sequences, boasting an impressive accuracy rate of over 90% in predicting disease-causing mutations. It can also design novel genomes that match the complexity of those found in simple bacteria. This remarkable capability positions Evo 2 as a formidable tool for expediting genetic research, with the potential to drastically reduce both the time and financial resources typically required for traditional experimental methodologies. Notably, the model has demonstrated a remarkable ability to evaluate mutations in the BRCA1 gene, which is closely associated with breast cancer, facilitating the distinction between benign and pathogenic alterations. One of the standout features of Evo 2 is the Evo Designer, a user-friendly interface that enables researchers to effectively utilize the model. The commitment to transparency is reflected in the release of Evo 2's training data code and model weights on GitHub, aiming to encourage collaborative efforts and innovation within the scientific community. Furthermore, the model's architecture has been enhanced through the development of StripedHyena 2, allowing it to process genetic sequences of up to 1 million nucleotides, significantly elevating its analytical capabilities when compared to Evo 1. Ethical considerations in development. The ethical dimensions of Evo 2's development have been a priority for the research team. They have taken measures to ensure that pathogens affecting humans were excluded from the training dataset, demonstrating a commitment to responsible AI deployment within biological research. This proactive approach underscores the importance of ethical considerations in the rapidly evolving landscape of AI in health and biology. Collaborative efforts and scientific community impact. The announcement of Evo 2 has been highlighted by various sources, including the ARC Institute and HPC Wire, emphasizing not only the model's monumental scale and capabilities but also the collaborative nature of the initiative. By providing insight into genetic codes and fostering innovation, Evo 2 is expected to play a pivotal role in the development of targeted gene therapies and various biological tools. Technical aspects of Evo 2's development. Creating Evo 2 required significant computational resources and innovative AI architecture. The model was trained over several months on the NVIDIA DGX Cloud AI platform via AWS, utilizing more than 2,000 NVIDIA H100 GPUs. The collaboration between researchers and engineers from NVIDIA was instrumental in achieving this feat. Greg Brockman, co-founder and president of OpenAI, contributed during his sabbatical to the development of StripedHyena 2, facilitating the model's ability to handle 30 times more data than Evo 1 and reason over 8 times as many nucleotides simultaneously. Potential applications of Evo 2. Evo 2 is already demonstrating its promise in biological research. Its ability to accurately identify genetic mutations that affect protein function and organism fitness opens avenues for significant advancements in medical research. For example, in trials with variants of the BRCA1 gene, Evo 2 achieved over 90% accuracy in distinguishing benign mutations from those that could lead to disease. This capability could potentially streamline genetic investigations, reducing the reliance on extensive and costly cell or animal experiments. Beyond genetic analysis, Evo 2 could also serve as a powerful instrument for designing new biological therapies. For instance, researchers could develop gene therapies that activate specifically in targeted cell types, thereby minimizing side effects and enhancing treatment precision. Hani Goodarzi, a co-author and computational biologist at UCSF, noted that such targeted treatments could revolutionize the approach to gene therapy. Future implications and opportunities. The research team envisions Evo 2 as a foundational model that could lead to the development of more specialized AI applications in biology. As David Burke, CTO of the ARC Institute, articulated, Evo 2 can be likened to an operating system kernel, paving the way for various applications built on its capabilities. The model's generalist understanding of the tree of life enables it to be utilized in a diverse array of tasks, from predicting the effects of single DNA mutations to designing genetic elements that behave differently across different cell types. Commitment to open science. In a significant move towards promoting open science, the ARC Institute has made Evo 2's training data and model architecture publicly available. This decision aims to empower researchers globally and facilitate collaborative efforts in addressing pressing health challenges. The foundation laid by Evo 2 is anticipated to ignite further innovations and applications in the field of generative biology. Conclusion. The unveiling of Evo 2 marks a pivotal moment in the intersection of artificial intelligence and biological science. With its capacity to process extensive genomic data and predict pathogenic mutations with remarkable accuracy, Evo 2 stands as a testament to the potential of AI in transforming genetic research and therapeutic development. The emphasis on ethical considerations, transparency, and collaboration ensures that Evo 2 will not only foster scientific advancements but will also uphold the integrity of research practices in the biological sciences. As the scientific community begins to explore the capabilities of Evo 2, its full potential in unraveling the complexities of genetic codes and developing innovative therapeutic strategies is just beginning to unfold. The announcement of Evo 2 represents a significant advancement in the intersection of artificial intelligence and biology. Here are some counterintuitive insights derived from the details presented. 1. Open source versus competition. While one might expect that a groundbreaking AI model like Evo 2 would be tightly held by its creators to maintain a competitive edge, the decision to make it fully open source is counterintuitive. This approach prioritizes collaboration and innovation over proprietary advantage, allowing researchers worldwide to access and build upon the model, potentially leading to unforeseen advancements in the field. 2. Ethics in AI training. The decision to exclude pathogens affecting humans from the training dataset might seem overly cautious in the face of scientific advancement. However, this aligns with a broader ethical imperative in AI development that emphasizes responsible use of technology. The counterintuitive aspect is that by limiting the training data, the creators enhance the model's applicability for safe research rather than risking misuse in sensitive areas. 3. Generalist model for specific applications. Evo 2's design as a generalist model capable of handling a vast range of biological inquiries stands in contrast to traditional approaches that often focus on highly specialized models. This broad capability enables it to perform tasks ranging from mutation prediction to genome design, suggesting that versatility can lead to efficiencies not achievable through narrowly focused models. For AI's role in accelerating traditional research, the notion that AI can replace traditional experimental methods might lead one to think that it could render empirical research obsolete. However, Evo 2's role is more complementary. It significantly reduces the time and cost of research, suggesting that AI can enhance rather than replace traditional methodologies. This perspective shifts the narrative from AI as a disruptor to AI as an enabler of scientific discovery. 5. Computational resources versus model efficacy. The massive computational resources required to train Evo 2 over 2,000 NVIDIA H100 GPUs may imply that only well-funded institutions can develop similar models. However, the open-source release of Evo 2 democratizes access to advanced AI tools, allowing smaller labs and institutions to leverage this technology despite limited resources. This counters the expectation that only the largest players in the field can innovate at the cutting edge. 6. Impact on gene therapy. The capability of Evo 2 to design gene therapies that activate in specific cell types may seem to suggest a straightforward path to personalized medicine. However, the complexity of biological systems means that practical application will likely involve unexpected challenges. This highlights a counterintuitive tension between the model's potential and the unpredictability of biological responses, suggesting that while AI can inform therapy design, it cannot guarantee outcomes. 7. AI as a biological interpreter. The idea that Evo 2 can read, write, and think in the language of nucleotides might suggest a level of understanding akin to human interpretation. This anthropomorphism can be misleading, while the model can identify patterns and make predictions based on data. It lacks true comprehension. This distinction is crucial because it underscores the limits of AI in biological contexts and the need for human oversight in interpreting results. 8. Interdisciplinary collaboration. The collaboration between various prestigious institutions and researchers from different fields, bioengineering, chemical engineering, computational biology, challenges the notion that groundbreaking innovations come from isolated teams. Instead, Evo 2's development reflects a growing recognition of the importance of interdisciplinary approaches in tackling complex scientific questions. 9. User-friendliness of complex models. The introduction of a user-friendly interface Evo Designer alongside such a complex model might seem contradictory. Typically, advanced AI models are associated with steep learning curves and high barriers to entry. Evo 2's emphasis on accessibility suggests that even sophisticated technologies can be made approachable, potentially broadening the user base and encouraging widespread application in research. In summary, Evo 2's launch not only signifies a leap in biological AI capabilities but also challenges several preconceived notions about the nature of scientific advancement, ethics in AI, and the interplay between open-source collaboration and competitive advantage. These counterintuitive insights could foster a more nuanced understanding of the future directions in AI-driven biological research. The unveiling of Evo 2 represents a monumental leap in computational biology, but several nuanced aspects of this development are likely to be overlooked by those focused primarily on the headline capabilities. Below are key points that merit attention due to their significant implications for the future of biological research and healthcare. 1. Scale of dataset and training methodology. Evo 2's training on over 9.3 trillion nucleotides from more than 128,000 genomes is unprecedented. The scale is not merely about the quantity of data but also the diversity it encompasses, covering all domains of life, bacteria, archaea, and eukaryotes. This diversity allows Evo 2 to identify patterns across vastly different organisms, thereby enhancing its predictive capabilities. Significance. The vast and varied dataset enhances Evo 2's generalization ability, allowing it to identify mutations and their implications across a broader context than previous models. This could lead to insights that are applicable not just to human health but also to agriculture, environmental biology, and synthetic biology. 2. Evo Designer, user-friendly interface for researchers. The introduction of Evo Designer, a user-friendly interface, is crucial for democratizing access to sophisticated AI tools. It allows researchers, regardless of their coding background, to interact with the model effectively. Significance. By lowering the barrier to entry, Evo Designer encourages broader participation in genetic research, enabling researchers from diverse scientific backgrounds to leverage AI in their work. This could lead to unexpected innovations as more scientists explore the tool. 3. Commitment to ethical development. Evo 2 was built with ethical considerations at the forefront, specifically by excluding pathogens that affect humans from its training dataset. This decision reflects a consciousness about the potential misuse of AI in biology, especially concerning bioengineering and genetic manipulation. Significance. This proactive approach to ethics in AI development sets a precedent for future projects. It emphasizes the need for responsible AI deployment in sensitive areas like genomics and could inspire similar frameworks in other fields of AI. 4. Interdisciplinary collaboration. The project is a collaboration among several prestigious institutions including the ARC Institute, NVIDIA, Stanford University, UC Berkeley, and UCSF. This interdisciplinary approach is essential in tackling complex biological questions that require diverse expertise. Significance: the collaborative nature of Evo 2's development could serve as a model for future scientific endeavors. It highlights the importance of pooling resources and knowledge across disciplines to foster innovation and address multifaceted challenges in biology and medicine. 5. Potential for tailored gene therapies. Evo 2's ability to design new genomes and predict the effects of mutations suggests it could play a critical role in developing targeted gene therapies. For example, the model could help create therapies that activate only in specific cell types, reducing side effects. Significance: tailored therapies could revolutionize treatment paradigms, particularly in complex diseases like cancer, where precision medicine is crucial. This capability may lead to significant advancements in how therapies are designed, tested, and implemented in clinical settings. 6. Advancements in understanding evolutionary patterns. The model's ability to detect encoded biological information in DNA and RNA reflects a deeper understanding of evolutionary processes. Evo 2 can identify patterns that have emerged over millions of years, providing insights into how organisms adapt and evolve. Significance: this understanding could lead to breakthroughs in evolutionary biology, ecology, and conservation efforts. It may also assist in identifying how certain genetic traits can be harnessed for better crop yields or disease resistance in plants and animals. 7. Open source commitment and community engagement. Evo 2 was the largest fully open-source AI model in biology, with its training data code and weights available publicly. This commitment to transparency is significant for fostering collaboration and innovation among the scientific community. Significance: open-source initiatives can lead to rapid advancements and iterative improvements as researchers around the world can build upon the work done with Evo 2. This could accelerate discoveries in genetics and related fields significantly faster than proprietary models. 8. Technological innovations in AI architecture. The development of the StripedHyena 2 architecture, which allows Evo 2 to process genetic sequences of up to 1 million nucleotides, represents a significant technological advancement. This innovation is crucial for understanding complex genetic interactions. Significance: the ability to analyze longer sequences opens new avenues for research into genomic architecture and function, potentially revealing previously hidden relationships between genes and their expression. 9. Foundation for future specialized models. The researchers envision Evo 2 as a foundational model upon which more specialized AI applications can be built. This concept of an operating system kernel for biology suggests a modular approach to AI in the life sciences. Significance: this could promote the creation of tailored models for specific applications such as personalized medicine, agricultural biotechnology, or ecological modeling. It supports the idea of scalable and flexible AI tools that can adapt to various research needs. Conclusion: while the capabilities of Evo 2 in predicting pathogenic gene mutations and designing new genomes are impressive, the underlying implications of its development are profound. The focus on ethical considerations, interdisciplinary collaboration, and open-source accessibility positions Evo 2 as not just a tool for genetic research but as a catalyst for broader scientific progress. Its potential to influence personalized medicine, enhance our understanding of evolution, and foster innovation across disciplines cannot be overstated. As the scientific community begins to leverage this model, the ripple effects on health, agriculture, and environmental science are likely to be substantial. 1. Breakthrough scale and capability of Evo 2. Evo 2 represents a monumental leap in the scale of AI models applied to biology, being trained on over 9.3 trillion nucleotides from more than 128,000 genomes across diverse life forms. This extensive training enables it to not only predict disease-causing mutations with over 90% accuracy but also to design new genomes akin to those of simple bacteria. This capability drastically reduces the time and costs associated with traditional genetic research, emphasizing Evo 2's role as a foundational tool for accelerating discoveries in the life sciences. 2. Open source commitment and collaborative potential. Evo 2's developers have prioritized transparency by making the model's code training data and weights publicly available on GitHub. This open-source approach is expected to foster collaboration within the scientific community, allowing researchers worldwide to build upon the foundational model. By providing a user-friendly interface called Evo Designer, the project aims to empower researchers to leverage Evo 2's capabilities effectively, potentially leading to innovative applications in gene therapy and other biological tools. 3. Ethical considerations in AI development. The research team has taken significant ethical precautions by excluding pathogenic data from the training set, reflecting a commitment to responsible AI deployment. This decision not only addresses safety concerns but also emphasizes the necessity of ethical considerations in the development of large-scale biological AI models. As the field progresses, ensuring that AI technologies are developed and utilized responsibly will be crucial to maintaining public trust and advancing scientific research ethically. 4. Foundation for future specialized AI applications. Evo 2 is envisioned as a foundational model upon which more specialized AI applications can be built. Its generalist understanding of the tree of life allows it to be a versatile tool for various tasks, from predicting mutations to designing targeted gene therapies. The innovative architecture, including the StripedHyena 2 system, enables Evo 2 to process long genetic sequences and reason over vast amounts of data, paving the way for future advancements in generative biology and personalized medicine. This potential for specialization highlights Evo 2's role not just as a singular advancement but as a catalyst for future innovations in the field. 1. The Human Genome Project (HGP), launched in 1990 and completed in April 2003. The Human Genome Project was an international scientific research initiative aimed at mapping and understanding all the genes of the human species. It was one of the most ambitious biological projects ever undertaken, involving the sequencing of the entire human genome, which consists of over 3 billion DNA base pairs. The project provided a reference genome for human beings and has significant implications for medicine, genetics, and anthropology. The data generated is publicly accessible, fostering collaboration and innovation in genetic research. 2. CRISPR-Cas9 gene editing technology. Developed in the early 2010s, CRISPR-Cas9 is a groundbreaking technology that allows scientists to edit genes with unprecedented precision and efficiency. This technology is based on a natural defense mechanism found in bacteria, which use it to protect themselves from viruses. Researchers have harnessed CRISPR-Cas9 for a variety of applications, including correcting genetic defects, treating diseases, and improving agricultural crops. The open-source nature of CRISPR technology has led to widespread collaboration and rapid advancements across genetic engineering fields. 3. The ENCODE Project Encyclopedia of DNA Elements. Initiated in 2003, the ENCODE project is a research project aimed at identifying all functional elements in the human genome. It seeks to provide a comprehensive understanding of the roles that various elements of the genome play in gene regulation and expression. The project has produced a wealth of data that is freely available to the scientific community, enabling researchers worldwide to study gene function and regulation in greater detail. The ENCODE project has been pivotal in understanding the complexity of the human genome and its implications for health and disease. The unveiling of Evo 2 marks a transformative moment in the field of biological research, with predictions indicating its profound impact over the coming years. As the largest open-source AI model in biology, Evo 2's capabilities in predicting pathogenic gene mutations and designing novel genomes present numerous opportunities and challenges for the future of healthcare and biological sciences. Enhanced disease prediction and personalized medicine. Evo 2's ability to accurately identify disease-causing mutations such as those in the BRCA1 gene is expected to revolutionize genetic diagnostics. Over the next decade, we can anticipate a significant shift towards personalized medicine where genetic testing will guide treatment decisions based on an individual's unique genetic makeup. This precision will facilitate earlier diagnoses of genetic disorders and cancers, enabling proactive interventions that can potentially improve patient outcomes. Accelerated drug development. The model's proficiency in understanding complex genetic data is likely to accelerate the drug discovery process. By utilizing Evo 2 to predict how different mutations affect drug responses, pharmaceutical companies could streamline clinical trials, reducing both time and costs. The integration of Evo 2 into existing drug development pipelines could lead to faster identification of viable drug candidates, particularly for complex diseases that have historically been difficult to treat. Innovations in gene therapy. Evo 2 opens up new avenues for designing targeted gene therapies. Researchers may capitalize on its capabilities to create therapies that activate in specific tissues, minimizing side effects while maximizing therapeutic effects. This could lead to significant advancements in the treatment of genetic disorders and cancers, with therapies tailored to individual patient profiles, thereby enhancing efficacy and safety. Broader applications in synthetic biology. The model's ability to design new genomes could spur innovation in synthetic biology. By leveraging Evo 2, researchers could engineer microorganisms that produce biofuels, pharmaceuticals, or other valuable compounds more efficiently. The future may see synthetic organisms designed for specific environmental applications, such as bioremediation or carbon capture, contributing to efforts against climate change. Ethical considerations and responsible AI use. As with any powerful technology, the deployment of Evo 2 raises ethical considerations. The commitment to exclude human pathogens from its training dataset indicates a proactive approach to responsible AI use. However, as the technology evolves, ongoing discourse surrounding bioethics, data privacy, and the potential dual use of AI in biology will be paramount. The scientific community will need to establish robust guidelines to navigate these challenges effectively. Collaborative research and open science. Evo 2's open-source nature promotes collaboration across institutions and disciplines. This democratization of access to cutting-edge technology is expected to foster innovation and accelerate discoveries in biological research. We foresee a future where researchers worldwide can build upon Evo 2, leading to diverse applications and potentially groundbreaking findings that address global health challenges. Evolution of foundation models in biology. Evo 2 is positioned as a foundational model for future specialized applications in biology. Similar to how operating systems enable various software applications, Evo may second serve as a kernel for developing more tailored models addressing specific biological questions. As the field of generative biology matures, we can expect a proliferation of specialized AI models that leverage Evo 2's insights, enhancing our understanding of the tree of life and its complexities. Conclusion. The introduction of Evo 2 heralds a new era in biological research characterized by enhanced predictive capabilities, personalized medicine, and innovative therapies. Its impact will likely extend beyond genetics to influence broader applications in synthetic biology and environmental science. However, navigating the ethical landscape and ensuring responsible deployment will be critical to harnessing its full potential. As researchers continue to explore the capabilities of Evo 2, it is clear that the future of biology will be increasingly interwoven with advanced AI technologies, transforming our approach to health, disease, and the environment.